Here was the picture, of them growing today. They've been around a long time. These are the modern day descendants of those very first fossils, still growing, possibly forming fossils because that's in the perfect environment for sedimentation to occur and all of that. Um, these stain as gram-negative rods, and when I get on into the bacteria here and talk about staining, you'll see why all archaea are going to stain as gram-negative, but they're really not. Um, that classification doesn't really apply. But it's one of those things, if you're looking at something and you're not sure and you stain it and it stains gram-negative, then it's like, oh well, it could be then. If it's looking like if it's looking like it's an archaea and it stains negative, it might be. Okay, because they just don't have the right thing to take a stain. So like I said, more on that in a little bit, probably later at the end of the hour maybe. Uh, we thought these were algae because they were so big in comparison to most bacteria. Um, they're going to have chlorophyll because they they photosynthesize, and you can't photosynthesize without having chlorophyll. That's a pigment required for that process. But they may, and a lot of them do, have pigments in greater quantity than the chlorophyll. So you'll see a different color when you look at them because that chlorophyll is masked by this larger quantity pigment. So they can be red, they can be yellow, brown, or black in color. And then talking about the size, um, the they're between 1 to 50 micrometers. Okay, so a micrometer is very tiny, but we can see it under magnification in a microscope. What I do when I'm at school is I pull a ruler out of my drawer and I turn it to the centimeter side, not the English inches, but the opposite side is in centimeters, and I'll have the students say, look here between numbers 1 and 2, and see that distance between 1 and 2, and that distance is marked into 10 smaller marks. Now look at the distance between those smaller marks, which is approximately what I got my fingernails here. You can see that on the screen, very tiny. Take that and divide that little space 1,000 times. Okay, a micrometer is a millimeter divided 1,000 times. Okay, so it's very, very small, but if you put that under a microscope, under oil immersion, which is 1,000 power magnification, you can see it. Okay, so um, but still, like I said, considered at 50 micrometers, that's considered kind of large for bacteria. Okay, we see single-celled organisms like algae in that range. Then when we got to where we could see down inside and see the organelles inside of that, then it became obvious that these things did not have organelles and therefore they weren't algae. A lot of our Archaebacteria are what we call extremophiles. They love extreme environments, phile being to love, lover. Okay, so we have things like halophiles. They love salty. Halogens are another name for the salt, kind of salts family. So halophiles, they love salt. Thermophiles, they thrive in very hot water, thermo. Okay, there is another term for cold. The ones that love cold, and we find those in the polar ice caps, Antarctic ice and Arctic ice. Um, a lot of the exploration that's been going on in Antarctica right now, or past 2 years, there's been talk about the cold-loving archaea that they've identified there, and some of this, I don't recall reading any new species identified. May have happened, but I just haven't read about it. But that the talking about the quantities of what they knew existed, but you know, finding more of it basically. Okay. Um, Methanogens live in anaerobic environments. They produce methane gas, and that's why they have that name. Okay, instead of a phile classification, so they're named for what they produce. We find these in swamps. We find them in our intestines, and they give off methane gas as a waste product. So some interesting facts about methanogens. Um, Scientists think that these are the bacteria that probably gave rise to the idea of ghosts, okay, spirits hanging around graveyards, because all organic material will decay, And methane gas has a couple of properties. It's kind of dense. It likes to, well, I'll say dense. Yeah, it's 5 carbons, but it's still, yeah, it's heavier than nitrogen gas and oxygen gas, which are made up of two atoms each. Okay, so yeah, methane gas is 1 carbon and 4 hydrogen, so five atoms. So anyway, it likes to stick to itself too. Okay, so it will kind of coalesce, and it will also phosphoresce in moonlight. It means it glows in the moonlight. Okay, so if you've got decaying organic matter, then and it's these methanogens are giving off this methane gas. Methane gas collects in a little cloud, and it's going to kind of hang low right above the ground, maybe carried around a little bit on air current if the air currents aren't too strong, because that would disperse it. Glow in the moonlight. Churches used to be in the middle of the neighborhoods. Okay, so people could easily walk to church. They weren't off somewhere on the edge of town or out anywhere, big. They're kind of in the middle of town. The graveyard was also right there next to the church, and you see this a lot back east. You see it in Europe. Okay, and not as much here, but yeah, the graveyard would be right there. So you've been over a friend's house, and now you're walking home at night, full moon out. You walk past the graveyard, right there above, headstone in front of it, above a grave, you've got this glowing ball of gas. Ooh, yeah. So, uh, because as the organic matter decays, if it does filter up through the ground, okay, and collect or can collect. So, also, um, lights in swampy areas. You know, swamp lights. It's been thought that the spook lights. We have our own spook lights, in Miami area, and it was thought at one time that that could be an explanation for that, but that one is not holding true. Okay. Um, Spook lights are. I used to run a summer science camp, and we take our kids out 2 different nights and do a spook light investigation. We took him to a spot where it was not spook lights, it was car lights, but um, they had to see it the first night and get all kind of nice and scared about it, and then we'd have them say, okay, what we take them the next day or during the day and have them uh, look at I think a map of the area, look at the area and figure out what could be an explanation. And typically, when they noticed that there was a road on the other side, they would say, "Oh, we're looking at car lights, maybe." And so then we'd split the next night into teams, and we'd take one van on one side and the other van on the other side, and flashlights and shining back and forth, and be like, "Ah, okay." So had the hypothesis and showed it, but that's not the explanation for every spotting of spook lights. It's just that's a location where you can go and quote guarantee to see them because it's car lights. Okay, so anyway, but that was this is like one of the scientific explanations for at least some of the kind of supernatural idea of ghosts around graveyards and things in swamps and stuff like that, is that this methane gas does have this property in our intestines. The methanogens try to help us out by digesting cellulose. Okay, I mentioned earlier that cellulose is one of those organic compounds that we can't get the glucose out of, but other animals can because they have multiple chambered stomach, or they have different, internal flora than we have. Internal flora, that's our intestinal bacteria, fancy word for that. Okay, um, and it's like, well, why don't we just pick that up too? Well, it's different species, different organisms can maintain different species of bacteria because of the circumstances in their guts being different. Okay, so these are the ones that we can maintain, and they only do kind of a partial job at being able to break that cellulose down. They break it down, they get some energy out of it for themselves. Um, you know, they can't break it all down for us. Partly because the matter doesn't stay in our intestines long enough. Okay, for them to do a complete job. So they do partial job enough for them to live. Uh, and like I said, get us maybe a little extra energy too. But the resulting of that is that we have gas. Okay, you eat those high cellulose foods, and it's not just cellulose. There's a couple of very large starches that occur in a lot of legumes, beans and peas and lentils, and those grouping of foods that are also usually highly gassy. And that's another thing. These try to break those down for us, and in most cases, actually, the big starches, they really do break them down and get food value out for themselves and us. But the price we pay is gas. So I refer to these as these socially embarrassing bacteria. There you go, or socially embarrassing archaebacteria to be precise. So now you kind of understand how that works a little bit too. All right. Um, I've talked about these as the extremists, the extremophiles, but they do actually have members of this group that live in normal environments. They don't have to be in these extreme environments. They don't necessarily, some of them can't live in the extreme environments either. They're going to just be moderate environments. But because they are the things, some of the only things that live in some of these extreme environments, that's often what they're known for and how we think of them first. Obviously, cyanobacteria is versatile. Okay, it can live in some extreme environments, and it can live in regular water. We have it down here in Grand Lake. A few years back, one summer, it got out of control completely. I mean, it just like took over and was causing issues. And Archaebacteria are typically not pathogenic. They don't cause human diseases. As what I've read, we have no known diseases caused by any type of Archaebacteria. However, people were getting sick from the cyanobacteria in the lake. And when I inquired about that, what were the symptoms? All I found was diarrhea. Okay, so that doesn't mean that they actually caused a disease in us. That just means people drank impure water. Okay, impure water. Um, because if you go to the ocean and you drink salt water, you're going to get diarrhea. Okay. The impurity creates a hypertonic situation, right? Okay, or hypotonic, depending on whether are you inside the intestines or outside in the cell. Anyway, water is going to get pulled out of your body. If you drink salt water, you drink impure water, it goes through your system, it's going to pull water out of your body, and that's going to create diarrhea. All right. So I think that's probably what was going on instead of cyanobacteria causing a disease. All right, so to speak. This is Grand Prismatic Spring in Yellowstone National Park. This was the spring that I studied. There were two. This was a graduate level class. There were two of us doctoral students, and she had everybody pair up. So we two doctoral students paired up and did a study, and we looked at the different types of archaebacteria here. These are all cyanobacteria. There's some pictures that are much better than this one, showing even a greater diversity of color. In the very center here, you can kind of see where my arrow is pointing. There's this really, really dark blue. And if you're looking down on this from an aerial, which there's pictures of people taken from the air, an airplane or helicopter, but you can also climb up on a bluff above this thing, and there was kind of a ways, but you can still get kind of an almost downward look picture of it. And when you're there standing, you can see that this area, this whole center is this really dark blue. That really, really dark blue, the temperature of that is boiling right at or right under boiling, and nothing is living there. Okay, it is a really that deep dark blue. It's reflecting the sky, but there's nothing there to cloud up that water, make it cloudy. So it's real clear and deep blue. Then you start seeing this kind of blue green appearance coming in. That's the first of the cyanobacteria, and it is a cyano, blue green. Okay, archaebacteria. And then that kind of fades into a kind of a mustardy yellow, and there's actually somewhere in there is a really bright yellow. You get a bright yellow, and then a mustard yellow, and then you get this orange, and then the orange gets into more of a brown and kind of a red. Okay, so there's a much greater spectrum in this than what you can see in this picture. Um, if you know the temperature range tolerance for your cyanobacteria, you can actually tell what temperature this water is. If you've got a chart there that tells you, okay, the cyano blue green is gonna live between this temperature and that temperature, then you can tell, well, here's where the boiling water drops to this temperature, and then from there it ranges from here to there because this next color takes over at that temperature, and this other one can grow maybe but doesn't like to grow at that temperature. It's more favorable for that warmer temperatures. So anyway, it's kind of one of those cool properties of these extremophiles. They got their range they really like, and that indicates to you what temperature that water is. Out here in this brown and red, um, you find little insects and things living out there. It's still warm water. Um, there's a kind of where this picture ends is probably a person was standing on the walk. There's a boardwalk they've got out there on solid ground. I don't want to go stepping off the paths, but um, that boardwalk, you walk out and be right over part of this brown red stuff, and I put my hand down right above the water, and you feel the heat coming off of it. Um, you can see the steam rising off this thing too. You can kind of tell a little bit in the picture that there's a little bit of steam rising, but out of the center. But it was warm, but it wasn't too warm. There was some insects and things that liked that warm water and were living in there. Okay. So it's a really cool place to visit if you ever get a chance, if you haven't been. I just always tell my students, I said, do be careful and stay on the paths. Okay, they caution people to stay on the paths because uh, this stuff forms a crust. You can see some of the stuff up back here in the back is a crust, and it looks solid and it's not. Okay, you can step on it, you just fall right through into possibly boiling water, near boiling water. It's happened. People have died that way, and it's not a pleasant way to die. Okay, if there is a pleasant way to die, that's not it. Being boiled to death is not what you want. Um, and it apparently doesn't take but a few seconds to do it because they've talked about people falling through and their friends are there to pull them out. But and yeah, they die soon after still because it's just your body can't handle that. All right, so stay on the paths. They put them there for a reason. Um, so the next article I want to talk to you about is this one. I let's see, this is up on your. I've got an article I need to add because I realized I didn't have it up this morning. Okay, but this is here, the thermophiles in Yellowstone.
So we're not going to have a test.
Oh, no, there's no quiz today. Thank you for asking. Yeah, I'm just not at a point in the lecture where, because we've missed so much and it's been kind of weird and disconnected. I haven't really covered to a point where I want to quiz you. I'm going to talk about aquatic biomes here, and that'll probably be possibly the rest of the hour after I get done with this. And that will be Monday. Okay, I'm going to I'm going to open that quiz up for Monday. So we'll have a Monday quiz. We may have a Friday quiz next week too, to kind of start balancing out that we've missed a few quizzes. But the the aquatic biomes are pretty focused. I'm going to tell you in a little bit really what to study for over the weekend. Okay. All right. So, but no quiz today.
Okay.
Yeah. Thank you for asking that. So this article here is about the Archaea in Yellowstone Park, and you can read through this, but what I want to emphasize here is the importance of this discovery for genetics and for what we've been able to do with the virus, okay, with the COVID-19. So in the 1960s, oops, I jumped around. There was a scientist named Dr. Thomas Brock who was out in Yellowstone investigating all these hot springs and these thermophiles that lived in them. And he isolated them out, looked at them, and he found this one in what's called the Mushroom Spring. Okay, one of the hot springs, not the one I just showed you, but another one. And he called it Thermus aquaticus. Thermus because of hot temperature and aquaticus water. Okay, so hot water. That's what this means. It's this bacteria loves hot water. Okay. Um, and what was important about this was at this time, in the 50s, Watson and Crick, right, had got the double helix shape. Well, we didn't stop there, of course. Geneticists kept going on. It's like, okay, now how can we get this thing to replicate itself? How can we replicate it? How can we mimic nature and make more because we want to and not just because the cell can? And one of the big roadblocks that we ran into in trying to be able to get DNA to replicate was that you know you can get it to separate, and we can get the nucleotides to come in there and line up, like I've shown you with replication. But then after that, we have to have what's called annealing, and the DNA, the two strands have to stick to each other and twist. Okay. And what happened was when we heat it up hot enough to separate, because we have to use heat and some enzymes, but we just don't. We're not as good as inside the cell. Okay, the cell can do the whole series of enzymes and processes and not have to use this high heat. The only way we could do it in a laboratory is use high heat to get those strands to separate to start with. But when we do that, we destroy the enzymes needed to get the new strands to stick together and helicize. Okay, so what do we do? We needed to find some enzymes that could handle that high heat that we were using to separate the original strands, and we didn't have any of those. But Dr. Brock finds this bacteria and says, hey, it is living at temperatures above 60 degrees Celsius, which was the highest we had then. And we have to put this up. And if I remember right, the temperature with the bacteria, with the, sorry, with the the viruses that I was working with in the switchgrass, I believe 72 degrees Celsius was our temperature that we heated up to split the strands. So these are thriving at between 70 and 80 degrees Celsius. Okay. Perfect, right? All right. Um, and so uh, later, it was a while. Okay, we have in '83, Dr. Carrie Mullis, okay, found that if he extracted out this particular enzyme. Okay, got to figure out. I mean, it's like we found the bacteria, but now we got to find the enzyme. What's the enzyme? We got to isolate that enzyme. We got to be able to utilize that enzyme. That took a lot of research and time. So it was, yeah, good 13 more years basically or so before uh, this researcher was able to um, isolate and identify this protein that was needed. And he named it TAC polymerase, TAQ for Thermus aquaticus polymerase. And of course, you've seen that term, I think, polymerase already in some of the diagrams for the DNA replication. Um, and so this was able to be used to make rapid copies of DNA in an apparatus that we call a PCR apparatus. And PCR stands for polymerase chain reaction. And what happens in this is you put the DNA or the RNA. I was doing RNA. The viruses I was doing were RNA, so it doesn't matter what you copy as long as you got the right nucleotides in there to make the right type of strand. And you can do DNA to RNA. Okay, just have you set it up with the right enzymes and all of that. But anyway, you put your template in there, and then you cycle it. You heat it up to break the template apart. You cool it down to allow other things to kick in and for the replication to happen, and for the um, uh, enzyme to, yeah, fix it all back up. And then you start the process over again. You heat it back up and you break those apart. And it's anyway, and you program the PCR apparatus to do this for as many cycles as you want to get as many copies as you need. It's called amplification. You have to increase these copies a lot, hundreds of times, in order to get enough to isolate out um, and actually work with anything, because it's too small for us to see. It's too small for us to pull out an individual thing. So we use multiple copies. So we're looking at multiple copies of the same thing. And that way we can see it. Okay. Um, so this was very important this last spring and summer in identifying the different strains of the COVID virus that was out there. We're still using it, obviously, to identify newer strains that are coming along, to sequence them, because in order to sequence, again, you have to have multiple copies of it in order to study it and be able to identify what those sequences are, what those actual sequences are. I have you draw it out in one, but we can't really work with it that way. Um, so uh, these apparati were in short supply in spring and summer, and in great demand. And the ones I worked with down at OSU, the building that I was in, the floor that had this, they had 4 of them, if I remember right. And you had to reserve them. Say, I'm using them at this time. Uh, but I understand that at least a couple of the researchers, if not maybe, I think just one led the way, but I think there were a couple other researchers that got together and they got involved in helping, okay, with the identification processes, and used the PCR apparatus at OSU. Okay, since there was kind of this call put out as if you've got one and can, help us out, because they want to do everything as fast as possible. Also, in my understanding, I'm not well read on the vaccine making process of this, but my understanding is that these PCR apparati are important, okay, in making these multiple copies of the messenger RNA that is in that vaccine. You have to make lots and lots and lots of copies of that, and to make the vaccine. So that's again kind of one of the bottlenecks probably on manufacture. Okay, is getting these copies replicated. But again, I don't know the full process that the pharmaceutical companies are using there. So again, what came out of a hot springs. So Yellowstone, as far as science goes, Yellowstone is still we haven't discovered, obviously haven't discovered everything out of there that we could probably, but it has been very valuable to science in trying to understand early life on the planet, understand these extremophile bacteria, and now applications to genetics too, and understanding um, genomes. Being able to map all that genome mapping that we've been doing for the past 30 years would not have been possible without Taq polymerase. Okay, it's a key discovery for us to be able to do what we can do with genetics. Okay, next topic, aquatic biomes. I don't have that pulled up. Must have accidentally closed it out. Okay, so time-wise, some people have argued that wars in the future might be fought over access to fresh water. What do you think? Think that's a possibility?
Yeah.
Yeah. Why? I'm always going to follow up with that, and you could always say, I don't know. I just felt like that's the right way. That's the right answer. But if you have a reason, I'm interested in knowing what your thoughts are.
Just because I've heard it, probably.
Yeah. It's not an uncommon theme in science fiction. Okay, that some futuristic society, and water's scarce for whatever reason. It's polluted, or the whole planet's dried, or you're on a different planet somewhere. I was saying that back in the 90s there was a movie called The Water World, and I think the water was everywhere, but none of it was really usable. And that's the problem. That's why we say fresh water. Okay, salt water, the ocean water, you've got to distill the salt out of that before you can drink it, or you get, like I was mentioning earlier, you get diarrhea, which will dehydrate you and kill you. So if that's all you drink, you're gonna die. Okay, you got to have fresh water. And let's see, my husband was telling me there's The Expanse is a series on Amazon that that's part of the source of conflict, I guess, is water and scarcity of water. I was talking about other planets in science fiction. Actually, Star Wars, the very first scene, first movie, I believe, is well, maybe not first scene, but anyway, Tatooine. Tatooine's a desert planet. What are they doing? They've got evaporators to pull water out of the air because there's just not. It's a desert planet. So yeah, not fighting wars over that necessarily, but still showing that it's important for life. We've got to have it. So why? List the reasons why fresh water is so important for living things. What do you think? What can you tell me?
We're electrical beings and we need it for conductivity.
Mm, yeah. Um, our nerves run off basically electrical impulses, and we have to conduct that across synapses. That current's got to be conducted across. So you've got to have that kind of that liquid. Um, all the chemicals that are used in all those metabolic processes I keep alluding to have to be dissolved in a solvent, right? And that solvent, that universal solvent, is water. Yeah. So, and all living things that we are aware of so far are like that. Do human societies all have equal access to fresh water?
No.
No, we don't. And that could be due to several factors. That could be due to just the people living in dry regions. or it can be due to maybe at one time having had more access to fresh water, but the water has become unusable for some reason, like pollution. I mean, we've had some water become radioactive because of certain accidents, to where you know
You have
lakes.
overpopulation is the biggest one.
Yeah, well, because that makes pollution, doesn't it?
Yeah.
Yeah, yeah.
We've outgrown our water source.
So we have to find ways of being better about not polluting, or cleaning up what we've already polluted if possible, and then trying to not add to it, and not really doing a good job of that. Okay, I mean, we got people working on it, and we're doing some, and we have done a little bit in certain aspects, but it's like we clean up one type of pollution and we'll create another type. It's just, yeah.
I always find it funny. We talk about we need to make, quit our dependency on fossil fuels. We need to build this and build that for green energy. I'm like, it takes even more power to grow to produce those than, yeah.
Yeah. So catch 22.
That's 22. Either burn the coal or burn the natural gas to run the furnaces, to run the production, to make the green stuff, or make the batteries, or at the mine for the lithium
Yeah.
and everything else.
Right. It's like, well, solar would be great, and yeah, I still think we'll figure that one out. I think we're figuring it out, and I think we're gonna figure it out someday. But right now, you're right. The batteries have heavy metals in them that they wear out, and when they wear out, what do we do with them? That's pollution, and the not just the batteries, but the photovoltaic cells themselves have a lifespan of like 10 years or less. Now, I understand that's expanding. They've gotten some better ones that can go longer. But again, you've got heavy metals in those that, what do you do with those when you get done? So it's like, well, the answer to that could be recycling, but then you've got to figure out that process. And and the recycling process is going to require energy and probably have some waste products. And
Actually, it takes more energy to recycle it than to make new.
well, yeah, there you go. And that's been an argument against recycling, is that no matter what you do, it's like more energy to recycle than new. But is it worth it? Maybe.
There's no good answer.
Because if you're recycling, you're reducing pollution. Even if it requires more energy, at least you're cleaning up after yourself, and we've got to do some of that. So maybe we need to spend that energy. Yeah, but where's that energy coming from? So yeah, wow. And that's why it's such a big issue. And why we need to put effort into trying to solve those puzzles, because we really don't have a choice. We're going to have to solve those puzzles at some point. And I think there's a solution. It's just, again, we may have to learn some more and discover some more things that we don't currently know or have in order to be able to solve those puzzles. But necessity is the mother of invention. So there we go. We put our minds to it, we'll figure it out.
So we just need to purge.
Purge. Well, that's one solution, but I don't think that's a good one. Don't think that's a good one. Some of those science fiction stories do that, yeah.
Yeah.
And so here's usually where I do talk about that. I got to get through this real quick here. There was another thing I just talked about with 9 o'clock that I skipped over with you. Okay. Freshwater biomes they're classified, any water body is classified as freshwater if it contains less than 1% salt by weight. Okay, 1% by weight salt. So that's like 1 gram in 100 milliliters is what that would be considered percent weight. Okay. We have our biomes here are lakes and ponds, rivers and streams, and wetlands. And this is in your book. This is back in the back, chapter 13, maybe. What's the last chapter of your book? First couple of pages, I think. There's a 2-page spread in there somewhere of all these biomes. Okay. Freshwater biomes cover less than 1% of the Earth's surface, but are home to nearly 6% of all species. So a lot of things try to live on just a little bit of water. Okay, and then in our marine biomes, this is where we have generally 3% salt by weight. We have coral reefs. We have the pelagic realm, okay, which is sometimes referred to as a zone as well, but it is actually broken down into some other zones. photic zone and aphotic zone. Photic means light, aphotic means without light. Okay. And that is further described in your book on that. Those zones there, I think, are emphasized more in the book than in the article I'm going to talk about here in a little bit. And then we have the deep sea. So, well, we talked about less than 1%. We talked about 3%. Well, that leaves this gap in between. What happened to the 2%? Okay. And that's going to occur where these two types of water meet and mix and you get this range of something 1% to 3%. These are called intermediate aquatic biomes, and they include the intertidal zone and estuaries. Okay, so that's the overview, the quick overview. And then I have this article in your modules for you to read about the aquatic biomes. Okay, and this breaks it down into more detail. And what how I ask this is these are multiple choice questions. Whoops. And a lot of it is matching definition of like these zones or the, what kind of, the larger biomes, what do you find in them? Because this article will tell you about what kind of animals you find in the different biomes. And then if you notice here, ponds and lakes. Okay, this is our freshwater, one of our freshwaters. I talk about, oh gosh, I should keep clicking on that wrong. Near the shore, we have what's called the littoral zone, and then out further in, we have the limnetic zone. Okay, and it talks about the different types of species and organisms you find there. The profundal zone. Okay, it's more a lake type thing. I don't ask you about temperatures in these except for when we get out to the deep ocean. And then I do have, I think that's the coldest waters. I talk about what's the coldest waters. Okay, streams and rivers and wetlands, those are details there on what lives in those other freshwater environments. And then here's the marine regions. And so talks a little bit about the intertidal zone there. Okay, it's one of the intermediaries. And what lives there. Here's the pelagic zone. This is what you hear. It's region in your book. And then this is divided into the aphotic and photic zones. Benthic zone, okay, is underneath the pelagic zone. And then we have the abyssal zone, which is the really, really deep part. Okay, and this is the coldest region, around 3 degrees Celsius. Okay, you don't have light down there either. So what you find is you find things like chemosynthetic bacteria that are going to break down chemicals for their energy instead of photosynthetic product like sugar. That's not going to be there because photosynthesis isn't occurring. And then when things die, they fall down. Okay, so you have scavenger things down here that do eat decaying things. Coral reefs are going to be in the shallow areas, shallow and warm waters. And these, of course, are a focus of conservation. There's places in the world where we've totally lost some coral reefs in that area. There have been efforts to try to bring back or catch before they're totally gone some coral reefs and try to restore some of the diversity that was lost. I've read that some of that's been somewhat successful, but it's kind of almost a stalling game right now instead of a permanent fix. Okay, and so this is an area of concern with a lot of scientists. Okay, marine biologists and environmental scientists are looking at when these coral reefs go, it destroys the whole area because so much diversity lives on these. But not just that, it's a lot of other things visit these areas, okay, for food. And so it really affects a larger part of the ocean than just the coral reef itself. Okay, and estuaries, I talked about there. Okay, so that's the bulk of your quiz on Monday. It's going to be off this article and out of your book. It goes along with those 5 slides I just presented. Okay. So to talk a little bit back on this Archaebacteria, the thermophiles, the cold-loving things, talking about outer space and science fiction and things today, made me think again. this is what we're looking for in outer space. This is really the type of bacteria we expect to find on Mars. They've got rovers there. Everybody's got rovers there right now, I think, and looking for that water, huh? Just landed one yesterday. Yeah. So everybody's looking for that water because right now we think everything needs water, right? We don't know life that doesn't require water. And if we can find the undersurface water, then we think we can possibly find some of these extremophiles and archaebacteria. Um, and we think it might be pretty similar to the type we find here on Earth if it exists there. It could be that we won't find any, but that doesn't mean we won't find life. And here's the challenge in science, especially in trying to find extraterrestrial life like that, is that we might not recognize it if it looks quite a bit different, if its structure and makeup is so much different from anything on this planet because the conditions on other planets are quite different. And I think Carl Sagan said that in his Cosmos film, there is that, if we don't know what we're looking for, we sometimes fail to see it. And that's happened right here on our own planet in history. Okay, we go back history of science, we have had things right under our nose and didn't recognize what it was because we didn't expect it to look like that. We didn't know what we were looking for, so we couldn't see it. Okay, once we had an idea of, oh, maybe it could look like this, or maybe it would do this process and we need to look for this process, and then we find this process going on and then suddenly there it is, and well, that's been there forever. Okay, but we didn't recognize it for what it was because we just didn't know what to want to look for. Okay, so we didn't see it even though it was in front of us, and that could be happening on Mars when we finally discover, that something is living up there. It may be something that we noted in the very first rover that was up there, but who would have thought that was a form of life, right? So we don't know, but right now the archaebacteria is what most of your astrobiologists expect that we will find on the planet, and I think they really do expect we'll find it. If we ever get a rover to the poles, that's frozen water on those poles, and so some of these cold-loving archaebacteria are probably there. That's kind of a really high expectation, is that we'll find archaebacteria like what live in the Antarctic ice here there. Okay. All right. That's where I will end today then. We will